Hi, it's Dr. Lewis again, and it's my great pleasure to introduce you to Dr. Jonathan Pevsner, who is a research scientist at Kennedy Krieger Institute and is also affiliated with Johns Hopkins Medical Institutions. His work in bioinformatics and studying the genome are fascinating aspects of his work, and I'm going to hopefully get him to connect it to what we do in our class in this interview.
This is a time of tremendous opportunity to learn about our genomes and how it relates to all different areas of life, including teaching. So it's a great time to learn about this and understand what the connections may be. Well, the first thing is the state of technology, which is that we described the double helix with Crick and Watson in 1953, and then exactly 50 years later, in 2003, the Genome Project was finished. It wasn't until 2007 that we had the first individual genome sequenced, and that cost about $80 million. And then, in the subsequent years, the prices come down to where now it's only about $1,500 for a genome. What this means is that hundreds of thousands of individuals are having their genome sequenced right now, and that includes the genomes of people who are able to learn effectively or not, those who have intellectual disability or not. And so we're learning about the relationship between our DNA and the way that we behave in the world. And this has a lot of relevance to teaching. Sure. Bioinformatics, most simply, is going to the computer to further biology research. And bioinformatics is a discipline in which proteins, genes, and genomes are studied through computer programs or computer databases. What that means is we might take DNA and sequence it using a machine. We start with a blood draw from a child, purify DNA, use a sequencing machine to sequence all the 3 billion base pairs of DNA we have. And then we need the tools of bioinformatics to analyze sequences and figure out what they do and what the significance is of variation in DNA. This is challenging. There are thousands of different kinds of disorders, and to date, there are thousands of examples of diseases or different kinds of disorders in which a single gene mutation has been identified. And often those are diseases that cause a very severe phenotype or something that's very discrete. Examples include cystic fibrosis, Huntington's disease, and when you come to autism, it's much more difficult, or ADHD or learning disorders, because in most instances, people who have those different conditions don't have a mutation in one gene that it's easy to find. Instead, there are probably many genes that are involved, and it makes it more difficult to say, this person who has a learning disability probably has a mutation in one specific thing. Instead, we're lagging far behind in our ability to relate our DNA to how people learn. I think that many different kinds of conditions we have are passed down from mom and dad to the child. And the way that genes are transmitted can be very complex. And we can get a richer answer by looking at a whole family and looking at the uncles and aunts, the grandparents, as well as the children. If there's a child who has a learning problem, for example, understanding the nature of what the siblings are going through also is really important. In some specific cases, you can do linkage studies where we take a DNA sample from every member of the family. And I've been involved in several studies that do this, in which we can actually map the gene in an individual and say where the problem is. The information right now is very limited in terms of what is helpful today to understanding how to be a better teacher or understanding how students learn. What we're doing in this conversation is thinking about the near future. I don't know how near exactly, but it is likely that, as I mentioned, hundreds of thousands of genomes will be sequenced in the very near future of the next 1 or 2 or 3 years. And so we'll be able to relate things better. The way that I would frame this is in terms of genotype and phenotype. The genotype is your DNA, and that is the collection of DNA that is present throughout your body, and that specifies your proteins and makes your cells what they are and helps to define who you are as an individual in combination with your environment and how you interact with the world. That's the genotype. The phenotype is what we see in the person who's interacting in the world, And the phenotype could be someone who is on either side of the bell-shaped curve, who is an exceptional learner, or perhaps someone who has intellectual disability and learning challenges. How do we connect the genotype to the phenotype? That's our challenge, and I believe that if you take 100 children with autism, then in the very near future, it will be possible to sequence all of their genomes and identify what the genotypes are, and say 3 of these children have a mutation in the same gene. We can ask, for that particular mutation, what kind of approaches to teaching are most effective? What are the outcomes over a 10-year period? What medications have been useful? And I think that parents will want to come together and understand the group that their child is part of. Today, we don't quite know those groups for the general public, for typical children we might encounter who have autism. But we do know that from the first studies, if you look at the DNA from 5,000 children with autism, we might find a subset of just 10 with the same mutation. That connection between genotype and phenotype is going to really illuminate what kind of condition these children have and what strategies might be best to help teach them. Your question is wonderful and again very difficult because I think we're a little bit early. I think that there's tremendous interest in the promise of genomics to teach us why we have different conditions. But today, a teacher who studied genomics would not find a lot of material that would help them, that they could apply immediately. And I think that's an okay thing to say. Mm-hmm. It's not that it's the truth, as I see the truth, of where we are today. What that means to me is we should try to understand how genomics informs the brain, and the brain has plasticity and many different strategies for teaching are available. The genomics today is not going to give us many answers. If you take neurofibromatosis as an example of a disorder, there are many thousands of children who have mutations in a gene called NF1 that causes it. We're trying to understand across the length of a gene, we have many different places where problems can occur. When we look at the children with neurofibromatosis, we find many different types of learning disorder. How do we correlate them? And that's the process that's happening. So I think that's a good thing for teachers to know that this can be done. But this, at the basic research level, continues to be difficult to make a really strong correlation between one and the other. At the same time, this is integral to what the nature of the condition is that each child has who has a learning disorder. So it's an important question. At the same time, I'm saying that for a teacher who's ready to learn about this area, it's good to know what the possibilities are for the near future. In practice, I talk to a lot of families in which, based on the conversation, they decide that they will have their child's genome sequenced because the cost has come down, and this can be done for either research or clinical purposes. And a lot of parents aren't aware that this is even an option to try to see if there are chromosomal or DNA changes that might explain why their child has a particular way of learning. I'm interested in educational resources, and I think that in particular, educational resources for big data. Big data is a phrase that refers to digital assets, as the National Institutes of Health calls them. If you have a study in which DNA is sequenced from a child, billions of base pairs of DNA are sequenced, and terabytes of data, which is pretty large amounts of data, are generated just from one individual. And indeed, 1,000 terabytes is a petabyte. Sometimes we even need to think of petabyte storage of data. And so it's important to teach the teachers and train the teachers. And one way to do this is to systematically gather and catalog educational resources that exist. That includes courses and workshops and seminars, textbooks, and many different media.
Interviews.
Interviews, absolutely. And so if this interview is available either within a course or if it were posted to be more publicly available, it would represent an example of a resource that a teacher should be able to go find. And so when you mentioned that teachers might need to be educated about genomics and what its promise is for teaching, where does a teacher actually go? I'd like to see a centralized index that is very user-friendly to let a person say, I am here, I'd like to map my position. I am a teacher, I'm not a biologist, and I would like to understand genomics and what its relevance is to me. And I think gathering that information will be a large project and a challenge, but one that will be very useful. I think there's a sense of genomics as being really important for human disease. And for severe diseases, of course, we want to know how to cure them. But more broadly, I think there's a tremendous interest in understanding how the developing brain works and how the developing mind works. And I think that we have a sense of optimism that genomics can inform this. So I think that this is felt in the public. It's felt from a parent who has a child with a condition. And I think that we don't yet understand the complex relationship between our genes and our behavior. But we know that there are answers there. Well, I think I'm one of millions of people who love Leonardo, and that he's a well-known cultural icon. I think people come to Leonardo knowing him as the creator of the Mona Lisa and the Last Supper, and perhaps the two most famous paintings in the world. And as you study him, you find pretty quickly that there have been thousands of books written about him, and so much is known. And so as a teenager, I became interested in him, and I've collected 700 or 800 books on him and read all aspects of his life. And what I find is that he integrated art and science very closely. He was self-taught. He didn't go to university. He felt very defensive about not knowing Latin and started teaching himself in his 40s. And he was a lifelong learner. Although books were something of a novelty in the late 1400s, he collected almost 150 titles and went and tried to read all that he could on different subjects. So in terms of what he did, he integrated his art and his science very closely, and he saw painting as a science. And I think that that's really fascinating. It's a very integrative view of the world, and it's something that we can all learn from. It's been suggested that science by its nature is something that is a cumulative and progressive enterprise. Science accumulates knowledge and it progresses over time. So we build up facts about math that 2 times 3 is 6. And then we can build from that algebra, geometry, calculus, and different disciplines. The arts, it's been suggested, don't have that kind of cumulative process or progressive process. That's one worldview, and another worldview is that science may be thought of as a small and interesting subset of art. I think that if we bring the arts and the sciences together, that's a very healthy thing to do. It's a very holistic thing to do, and that it fits with the way the human mind works. It's in terms of education, I'm all in full support of arts being emphasized as strongly as the sciences. That information might consist of newborn genome sequencing in the near future, in which we can predict, based on the mutations that are seen in a child that's born, we can predict what learning challenges they may be having in the future and initiate earlier behavioral interventions. At the DNA level, we can look at something like Down syndrome, where there's an extra copy of a chromosome, and typically that extra copy comes from mom over 90% of the time. And children with Down syndrome have a huge range of outcomes in terms of intellectual disability or not, and how they're affected. And so we see great variation there. As we understand what's happening, we can see that some children with Down syndrome have particular gifts, and we see some overlap between Down syndrome and autism, where autistic savants are a well-known phenomenon.
Yes. Of
children having particular skill sets. I'd like to mention one specific disease that causes intellectual disability. It's called Sturge-Weber syndrome, and it's associated with a birthmark that occurs over the face. These birthmarks are very common. About 1 in 300 people have them, or over 1 million people in this country. And a subset of people with this common birthmark have intellectual disability and a disease that affects the brain. And we found the mutation that causes this disease by genome sequencing. We went into the affected part of the body, sequenced the DNA, and then looked at the unaffected part of the body from the same person and compared the genomes, and found that in the birthmark there was a mutation where 1 base pair out of 3 billion changed. So we could spot that, and we know what it does. We know that this piece of DNA normally makes a switch that is either on or off, and that in this condition that causes intellectual disability, the switch is slightly stuck in the on position. We could immediately look for drugs that can turn it back off. And we're hopeful that that can serve as a treatment for something that causes intellectual disability. So that's a happy example of a disease where finding a cause could actually lead to a treatment that's helpful. But also, it's a kind of warning of how complex the situation is.
Yes.
Because mom and dad don't have this mutation. It happened like lightning striking while the fetus is in the womb. And it happens by what we think is just bad luck. And when it happens, it's not something you could find by taking a blood draw and sequencing the genome. It's only in a particular part of the brain that the mutation occurs. And so nature can challenge us in lots of ways. And we think one family at a time, we can identify what those mutations are, if they exist, that might be causing problems such as intellectual disability. And I think that the prospects are great for going and doing that. We don't ever want to make the promise that finding the gene mutation can lead to a treatment or can lead to a better outcome for learning. But this does feel like the right direction to go. So I'm very interested in extending genomic studies to the population of students.
Well, thank you so much for giving me your time today, Jonathan. I think we're all going to learn a lot from this, and it's been delightful. Thank you.
My pleasure. Thank you.